Okay, hi everyone, I'm Damla. So right now I'm a staff software engineer focusing on hardware acceleration at Bionanogenomics, but I have graduated from Carnegie Mellon from the software research group of Professor Onur Mutlu last August. And today I will be presenting some works from my PhD thesis on accelerating genome analysis via efficient hardware algorithm co-design. So It's not working. I don't know why it's not working. Okay, thank you. Okay, so we went over some background today in multiple talks, but I will quickly go over some of them again. So genome sequencing enables us to determine the DNA sequence of an organism, and it plays a pivotal role in areas such as personalized medicine, outbreak tracing like COVID-19, or understanding of evolution. So, however, the current sequencing machines cannot take the full DNA sequence as the input and produces the whole thing as an output. Instead, it breaks it into smaller DNA fragments, and then they are randomized. So we lose the location information with respect to the original sequence. So that's why it is challenging. So when we look at the, from start to end, how it works, we start with a sample collection, get the DNA molecule out of it, and it goes through a wet lab preparation process, and then we extract those chopped DNA fragments. And then we feed these into sequencing instruments, like you are hearing names today about the companies producing these technologies, and then they produce those sequence reads, which are those randomized smaller fragments. And in order to get the meaningful information that we would want out of this process, then we have to go through a process called genome sequence analysis. So when we look at the range of technologies, as I said, we've heard some of them today, like Oxford Nanopore, PacBio, or Illumina, and they all produce different types of reads, which we can categorize as short reads or long reads. But short reads are a few hundred base pairs, but they are accurate in terms of their error rate. Whereas when we look at the long reads, they can reach millions of base pairs, but they are noisy. So again, another familiar picture. So as we know, the rate of producing genomics data and the cost of it is getting, the rate is increasing, cost is lowering, and it is exceeding the Moore's law we are observing, which would mean that the computation is becoming a bottleneck. So we cannot match the rate of compute analysis with the rate of generating the genomics data, which is a problem. And because of that, as part of my PhD work, our goal was to accelerate genome sequence analysis by efficient hardware algorithm co-design. And we followed a three-step approach for this. First, we analyzed the existing tools for the multiple steps of the genome sequence analysis pipeline and exposed the trade-offs between accuracy, performance, memory usage, and scalability. And after detecting the bottlenecks, we co-designed fast and efficient algorithms along with scalable and energy-efficient customized hardware accelerators for the key bottleneck steps of the pipeline. So my thesis covers 4 different research contributions. Today, I will be focusing on GeneAsm, which was published in Micro 2020, and also SIGRAM, which is published in this ISCA. But before jumping into GeneAsm, I also want to provide a very quick slide from our first paper, which Onur also mentioned this morning. So we revealed that based on our analysis with the existing tools that time in 2018, we revealed that we have to have high performance, low power, memory efficient, and also scalable, highly parallel solutions in order to enable fast and accurate genome analysis. And with that, I will start with our the first work for today, which is GeneAsm, which is an approximate string matching framework for genome sequence analysis. And before digging deeper into GeneAsm, I want to start with the read mapping pipeline. Again, we've heard it multiple times today, but both of the works we will cover today, both GeneAsm and SIGRAM, is focusing on the read mapping pipeline. So it's important to understand the steps So typically read mapping is following a seed and extend phase approach. So it has 4 steps. The first step, indexing, which is a pre-processing step. So it can be done only once for each reference genome and can be reused for multiple mapping executions. So we can treat it as the 0th step. So in there, we get the reference genome and create a static hash table-based index where the keys are the k-length substrings out of the reference genome, which are called k-mers. And the values for each key is the exact match locations of these k-mers within the reference genome. So as it is mentioned, reference genome is static. It is not changing very frequently, so we can generate this hash table once and reuse it. And then the first main step of seeding is getting the query read as its input. Again, finds these k-length substrings, k-mers, this time out of the query read, and then with each of these k-mers, go to the pre-processed hash table index and finds the potential mapping locations. Why they are potential? Because they are just the exact match locations within the reference genome, and it is just k-length substrings. So it's not covering the full query, which can reach millions, as we said. So then we have our potential mapping locations, but before sending them for read alignment, which is the most expensive step of this pipeline, we can have an optional pre-alignment filtering, or it's sometimes called as chaining or clustering based on the approach. In this step, the goal here is to get the reference segments from the second step and the query read itself and see whether they are similar enough to send for the read alignments. But if they are not similar enough, so they are dissimilar enough, then we can discard them and not send them for the alignment step. And with this optional step, as I said, we can get the inputs directly from seeding, but this helps us to decrease the burden on the alignment step. So we get the remaining potential mapping locations for the read alignment step, and again, get the pairs for the reference segment and the query read. This time performs a typically dynamic programming-based alignment approach where we find the distance or score for that alignment and also perform traceback in order to find the actual optimal alignment. So, as I said, read mapping is one of the very first key steps in genome sequence analysis. That's why we are focusing on this problem in these works. And since, again, mentioned multiple times today, we need to account for sequencing errors, which the devices can introduce, and also genetic variations we all have. The multiple steps of read mapping require approximate string matching instead of exact string matching. But based on our analysis, we revealed that approximate string matching, and not just our work, this is like a lot of works covered this, approximate string matching is one of the key bottlenecks of genome sequence analysis pipeline because of the computational power and memory bandwidth limitations of existing systems. So when I say approximate string matching, what I mean is, let's say we have these example pairs for reference and read. We need to be able to support any existence of deletions, substitutions, or insertions. So approximate string matching handles these type of errors. And it detects the differences and also similarities between two sequences. And in genomics, we require it because we want to find a minimum edit distance when performing alignment. And also with the traceback step, we also want to find a sequence of matches, substitutions, insertions, and deletions along with their positions. And as I said, ASM, which is approximate string matching, is typically implemented using a dynamic programming-based algorithm. But it has quadratic time and space complexity, thus we need different algorithms to solve this complexity or computational issues that comes with it. So in this work, we focus on BiTAP algorithm. So this is an old algorithm coming from the 90s, but it was never used in for approximate string matching in genomics, and the reasons we will cover. But we picked BiTAP because it only has fast and simple bitwise operations, making it extremely amenable for hardware acceleration. And since it's it is an ASM algorithm, it computes the minimum edit distance between a text and a pattern with a maximum of k errors. So one detail here is we have to define the k errors. That's the threshold we want to tolerate. And it has two steps, one pre-processing step where we convert the query pattern into its binary format by generating pattern bit mask for each character in the alphabet, ACGT in our case. So we generate 4 pattern bit masks per each query read and use it throughout the main step of the algorithm, which is the second step, which can be called as searching or edit distance calculation, which the actual action is happening. So here we compare all characters of the text with the pattern by using the pattern bit mask generated in the first step, and the status bit vectors that hold the partial matches between the text and the pattern, and also pure bitwise operations. So I won't be able to go into the more details of the algorithm, but just to highlight the limitations of the baseline BiTAP algorithm, I will show some pieces of it. So we detected 5 limitations of BiTAP, and the first one is there is 2-level data dependency within the algorithm. So what I mean is, this is a pseudocode of the algorithm, but I will just, as I said, focus on some portions of it. So there are 2 loops. In the outer loop, we iterate over each character of the text, and in the inner loop, we iterate over each possible edit distance, 1 to k. And when we consider long noisy reads, both of these loops, because the length will be higher, the outer loop, and because the errors will be high, the inner loop can be large. So we are dealing with large number of iterations. And on top of that, for both of these inner and outer loops, there is data dependency, meaning that, for example, for the inner loop, when calculating deletion, substitution, and match bit vectors, we have to get the data from the previous inner loop iteration. And also, for the outer loop, when calculating insertion, we need to get the data from the previous outer loop iteration. So we are dealing with data dependency for both levels of the loop. So on top of that, the second issue is there was no support for performing traceback, so finding the optimal alignment in the baseline BiTAP algorithm. So in the core of the algorithm, we are generating 4 bit vectors, deletion, substitution, insertion, match, which really hold the information we need to perform the traceback. But there was no algorithm to really traverse them and find the optimal alignment. So this was another limitation. And third, since we are each bit vector has a length equal to the query pattern. And when we think about CPUs where we are only limited by 64 or let's say 512, when we exploit the AVX kind of platforms. So we have a limitation in terms of query length because the bit vector has a length limitation unless we apply some optimizations to that. So the baseline BiTAP algorithm did not have any support for long reads, which can exceed 64 or 512. So these are the algorithmic limitations of BiTAP. And even if we somehow resolve these issues, we reveal that there are also 2 hardware-related issues. First of all, let's say we apply text-level parallelism, we divide the text into multiple overlapping chunks and perform the BiTAP algorithm in all of them in parallel. When we perform this on the CPUs, we find that we are limited by the number of compute units in existing systems. So that's why we switched to GPUs. But reveal that in the GPU scenario, the high memory bandwidth requirements to read and write the computed bit vectors was becoming the issue. So we were limited by memory bandwidth. And these are the hardware limitations of the baseline BiTAP algorithm. So obviously we need a completely new design, which is thinking from both algorithm level and hardware level in order to resolve these issues. So that's why we propose Genome. So in Genome, our goal is to accelerate approximate string matching by designing a fast and flexible framework, which can ultimately accelerate multiple steps of genome sequence analysis because ASM is used in multiple steps of genome sequence analysis pipeline. And Genome is the first ASM acceleration framework for genome sequence analysis by handling multiple use cases. And also it is the first work that utilizes BiTAP, modifies it, extends it, and make it suitable for being used in GSA. So we overcome all those 5 limitations that hinders BiTAP's use in genome sequence analysis, first by modifying and extending the BiTAP algorithm, then providing a novel bit vector-based algorithm that's compatible with BiTAP on top of the modified version of the BiTAP. And these cover our software contributions for Genome. And third, for both of the algorithms, we design a specialized low power and area efficient hardware. And this covers our hardware contributions in Genome. So when you look at the Genome hardware design, it's composed of two main components. First, Genome DC, which handles the distance calculation step, so the modified BiTAP algorithm step, so the hardware component corresponding to that. And second, Genome TB, which performs a traceback operation based upon our novel BiTAP compatible traceback algorithm. And we also have two types of on-chip SRAMs. First, DC SRAM, which is holding the input information required for DC operation, and TB SRAMs, which holds the bit vectors required for the traceback operation. So, excuse me. Let's look at the overall flow of Genome. So we start with the host CPU providing the reference and query location information to the Genome. And then Genome DC fetches this text and query that we will perform the approximate string matching operation into DC SRAMs. And in Genome, what we do is we follow a divide and conquer approach in order to reduce the computational complexity plus decrease the memory requirements of the algorithm. So what we do is we divide the text and pattern into multiple overlapping windows and operate on each window and then move on to the next one. So, and then eventually we combine everything. So because of that, DC first divides the text and pattern into subtext and subpattern and starts doing the modified BiTAP algorithm with that, generates bit vectors, and then writes them to TB SRAMs. Once DC execution is done, then TB starts reading these bit vectors back from the TB SRAM, finds a traceback output for the current window, and sends the information back to DC so that it can start with the next window. And it goes until all of the windows within the actual input, query, and reference region is done. So our specialized compute units and on-chip SRAMs, as I explained, help us to first match the rate of computation with memory capacity and memory bandwidth. Second, achieve high performance and also power efficiency. And third, scale linearly in performance with the number of parallel compute units that we add to the system. So let's quickly go over the Genome DC hardware design. So we implemented as a linear cyclic systolic array-based accelerator, which is helpful for maximizing parallelism and minimizing memory bandwidth and footprint. So this is the processing core, which handles one iteration of the inner loop of that BiTAP algorithm. So when we add the required flip-flop-based storage around it, we define a processing element, and multiple processing elements are concatenated so that we can define a processing block. And we add the DC SRAM that feeds the input and gets the intermediate data out of it, and also TB SRAMs, which holds the intermediate bit vectors. And when we look at the TB hardware design, it's even more simpler. So it only requires first reading the bit vectors from one of the TB SRAMs and then performing the required bitwise operations. So we are basically reverse engineering what DC did, but not for all iterations, but just for the optimal alignment path. So we reverse, do the reverse bitwise comparisons to find the traceback output for the current position. And then based on that found traceback output for the current one, we calculate the next TB SRAM address and reads the new set of bit vectors and go back to step one. And when the traceback output is found for the current window, then we stream it back to the main memory as the output for the current iteration. So because Genome is a flexible framework, we analyze three use cases of approximate string matching in genome sequence analysis pipeline, the read alignment step, the pre-alignment filtering specifically for short reads, and also since BiTAP plus Genome is an edit distance calculation algorithm, that's our third use case. And we also discuss other possible use cases of Genome in the paper, including generic text search when we are dealing with a larger alphabet than just ACGT. So because of time, I won't go into details of our evaluation methodology, but you can always refer to our paper or we can discuss later offline. And I will just show some key results from Genome. First, for area and power. So we can say that Genome is highly efficient in terms of area and power usage. So for all And one detail here, which could be helpful. So we model our main memory as an HMC kind of memory where we have multiple independent vaults. So we can put our logic into the vault and have a dedicated portion of the memory. And it will provide us like better low latency memory access without any interference from other like accelerators. So we have 32 volts in the model system. So we have 32 accelerators in total in the full Genome design. So when you look at all of them, it's both in terms of area and power, they are just taking 1% of SD on CPU core. So we can say that it has low area and power overheads. And just to quickly go over some key results we have for all three use cases. For all scenarios, we compare with state-of-the-art software and hardware baselines. For example, Minimap 2 and BWAMM, or Darwin and Genex when it comes to hardware, and for pre-alignment filtering, SUJI, and for edit distance calculation, eDelib as a software and ASAP. And for all the use cases, we show that Genome is providing better throughput. And if we have the power information available, less power than the baseline tools. So if you don't have any questions, I will quickly jump to SIGRAM.
7 minutes.
Okay. Okay, these are the additional details that you can find in the paper. And also, okay, I won't summarize them, Genome, I will quickly jump. So this is our paper. And one detail, our source code for the software implementation of Genome is also available in our GitHub page. Okay, SIGRAM. So as we said, read mapping, or in this paper, it's called sequence mapping, and you will understand why. It's a critical step. But we are using a linear representation of the reference genome. So it's just one genome you can take. But as we are talking about variations, that single reference genome cannot represent all the variations that we know, let's say for human, right? So because of that, recent works replace the linear reference sequence with a graph-based representation of the reference genome, which is called genome graph. And this kind of data structure captures the genetic variations and also diversity across many individuals in a population. So we are not getting stuck with just a single reference genome, which is causing reference bias for the downstream analysis like read mapping. So as we can imagine, we still need to perform mapping because it is a critical step and we have to do it. But now we, instead of a linear one, we have a graph-based one. That's why this is called sequence to graph mapping this time. And we know that it results in notable quality improvements in genome sequence analysis because it's more comprehensive. It's gathering more information. And that's why it's more accurate, as we can say. But we are dealing with a graph structure right now, so it's a more difficult computational problem, and it's a newer domain, as we can say. So there are much smaller number of practical software tools currently available, and there is no prior hardware design for graph-based genome sequence analysis. So we will propose the first one. So to understand what genome graphs are, let's say this is our initial sequence. So we have only a single node. But let's say we have another sequence in the population which has a variation on the fourth position. So instead of a T, we have a G. So this is a known variation, let's say. If you stick with a single linear reference, we would like label that G as an error and maybe correct it and move forward, whereas it was a known genetic variation. So we would be losing information when we could exploit it. So what we can do, we can divide the initial node into multiple nodes, have one path for the first sequence and have another path for the second sequence. So now we are representing both of them. Let's say we have a third one when there's an inserted T. Then we can have an inner node which has that T, and then now we have all these three sequences. Let's say we have a fourth one when that T doesn't appear at all, so there's a deletion. So we can have a direct path from the first node to the last node, and we managed to represent all those four sequences in a compact way in this genome graph. So I talked about sequence to graph mapping pipeline. As you can see here, it has a very similar, like it also follows the seed and extend strategy, but on top of what we had as indexing, seeding, filtering, and alignment, we need to also first construct the genome graph. But similar to indexing, it's a one-time step. So because the reference is almost static, so we have to like We don't have to reconstruct the graph, re-index it multiple times. We can pre-process them, so that's why we are labeling them as offline steps, and can reuse the data structures multiple times in multiple mapping executions. So since I explained the terms, I won't go into details for this one because time is limited, but as I said, we can talk offline, or you can refer to slides or the paper later. So one important thing to highlight here, for sequence graph alignment, it also typically follows a dynamic programming based algorithm, but what is different here is on the, okay, probably you cannot see it. Okay, anyways. So on the left-hand side, as you can see here on the sequence alignment, traditional read alignment, when calculating one score for the current cell, we only deal with the 3 neighbor cells, right? It is always like that. But when we move on to sequence to graph alignment, yes, we still have to consider those 3 neighboring cells, but on top of that, we might have hops, right? The edges coming from non-neighbor cells. And they can be random. We don't know beforehand. So it's not like regular. We don't know the distance between the previous node that the current node has, or even whether it has or not. So that's why we might have like edges coming from like non-neighbor cells. So we have to incorporate the scores coming from that column as well. So that's the additional complexity on top of the already expensive alignment step. So we analyze the existing sequence to graph mapping tools to reveal all the issues with them. So just to quickly go over them, again, because of time, we have 4 observations on the
2 minutes.
Okay.
Yeah.
So again, alignment step constitutes a majority of the end-to-end execution time, and it's even more expensive because of the additional complexity we add with the graph structure. Alignment suffers from high cache miss rates because we have a lot of intermediate data we are generating and reusing. So we have to put them in a cache, but if the cache is not large enough, then we are seeing the high cache miss rates. On the seeding part, because we are performing a lot of irregular memory accesses to fetch the seeds, locations, it suffers from the DRAM latency bottleneck. And as the fourth one, both of the tools scale sublinearly, so they cannot exploit the full hardware level like thread the opportunities we have on the hardware. So that's obvious that we need better algorithms with better hardware to support these. So we move on to some possible hardware accelerators. First of all, we looked at existing sequence-to-sequence mapping accelerators because we didn't have any sequence-to-graph mapping one. But we found that they are unsuitable for the sequence to graph mapping problem because we have to incorporate hops. And we also look at existing graph accelerators because we have many of them. So they could help the seeding step with the memory access part. But when we look at the alignment, which is the expensive part, they are completely different algorithms. They are completely different computational problems. That has nothing to do with graph traversal, for example. So existing graph accelerators are unable to handle alignment as well. Because of that, we propose SIGRAM. And in SIGRAM, we can say it's the first universal genomic mapping accelerator that can support both sequence to graph mapping and also sequence to sequence mapping for both short and long reads. And the reason is that we can treat sequence to sequence mapping as a simpler special variant of sequence to graph mapping when we all have like all the nodes are like linearized and only have one incoming edge. And this is also the first algorithm hardware co-design for sequence to graph mapping problem. When we based SIGRAM upon a minimizer-based seeding algorithm, and minimizer is a sampling approach for the seeding step. And also we propose a novel bit vector-based alignment algorithm, which is an evolved version of GNASM, which is handling the sequence to graph alignment. And we co-design both algorithms with high performance, scalable, and efficient hardware accelerators. So how long do I have?
Actually, we are over time.
Over time?
Yeah.
Okay.
We need to wrap soon because we need to take a break.
Okay.
Yeah.
Okay. Got it. So can I just go over this?
Yeah.
One more minute. Okay. Sure. So when we look at SIGRAM, it is also composed of 2 steps: min seed for the seeding part and bit align for the alignment part. And we pre-generate the graph-based reference and index preload to the main memory, and the execution starts when the host CPU streams the query reads. Then we, for each of those k-length substreams, again k-mers, we find the minimum representative set of them, the minimizers, write down to minimizer stretch path, fetch the occurrence frequencies of those minimizers from the main memory using the hash table-based index, filter the minimizers by the frequency so we can pick the least frequent ones, and then this time fetch the actual seed locations of them, write down to seed scratch path, and in MinSeed, find the regions that correspond to these locations, the actual subgraphs that we will perform alignment, and write them to the input scratch path. This is what MinSeed is responsible for. And then, as you see, we don't have any filtering approach because we rely on our efficient beta-line accelerator, but that's definitely a future work which can be explored. So then beta-line starts. So it's, as I said, it's built upon Genome. So again, for performing approximate string matching with graph structure this time, it generates bit vectors again. But different than Genome, we also hop queues in order to handle the hops. So we store the X amount of most recent bit vectors from the previous iterations, so we can handle the previous non-neighbor iterations to handle hops as well. But similar to Genome, we write down the bit vector scratch pad again because we need to perform traceback anyways. So after we perform traceback, we stream back the optimal alignment information back. So I won't be able to go into these details, but this is MinSeed hardware. This is beta-line hardware, which is very similar to Genome one. Again, we model our main memory as an HBM memory. So we have multiple accelerators. So one more highlight here. So Sigram is highly modular. So we can run it end-to-end to perform mapping, either sequence to graph or sequence to sequence mapping. But also beta-line and MinSeed can be standalone accelerators to perform their specific steps as well. And we evaluate all of these use cases with the baseline tools and reveal that Sigram or beta-line can increase the throughput for all of the baselines. And also, again, when we have the power information, we can decrease the power consumption with Sigram as well. There are much more additionals in the paper, which I couldn't include today in my talk. Here's our paper. And again, very last sentence, we will be releasing our software implementations and also the data sets we use in our GitHub page soon. Thank you.
Thank you. When is your talk?
The talk is on Tuesday, 3:10 p.m.
So if you ever miss any details, please come to the ISCA, to the main conference. Damla is going to be presenting full length.
Yeah. So for the rush.
I'm sorry. Questions? No, I was about to disconnect you.
Yeah, I'm about to disconnect.
But, sure, but if there's no questions? Oh, yeah, sorry.
Yes, so first of all, congratulations on the huge job, because building a software is hard enough, so I cannot imagine how hard it is to do this with hardware and accelerated. My question would be, how do you understand what is the maximum error parameter when you configure your accelerator and your running? Can you give us some examples of how does this impact accuracy?
Of course, that's a really good question. And as Nur was mentioning today, we use some in Genome, like accuracy.
Exactly.
This is coming from
You don't need to connect it.
it's coming from there.
I'm sorry.